Welcome to Ask the Expert. It's a brief, informative, and lively discussion with experts in type 1 diabetes and related interdisciplinary research. We're recording the event. We're going to post it later on our Sugar Science YouTube channel. And if you have any questions for our guests, please feel free to add them as we're speaking in the chat, and we'll answer them at the very end. Or if something's pressing, raise your hand and we'll answer it right away. Okay, so today I have with me Dr. Cassie Robertson, coming to us from UVA. And she, along with her colleagues, just published a really fascinating Nature Genetics paper. I have been sort of going over it and really enjoying it. I love that they went back into data, the title of the and found some really interesting things there. The title of the paper, which was published on June 14, 2021, is Fine Mapping Transancestral and Genomic Analyses Identify Causal Variants, Cells, Genes, and Drug Targets for Type 1 Diabetes. And so welcome. I can't wait to talk about this paper and your work. And again, and congratulations on your shiny new PhD. So
thank you.
So why don't you take a minute and give us a little bit of a background, a bio about yourself?
Sure. Yeah. So I just finished my PhD last week, actually. So my background is originally in statistics, biostatistics, and kind of has migrated more towards genetics. So first statistical genetics, and now really in kind of in the weeds of genetics. And the past 5 years, I've been working with Steve Rich and his group at the University of Virginia studying type 1 diabetes. And in January, I'll start in Steve Parker's lab at University of Michigan, also continuing to work in the genetics of type 1 diabetes and also a little bit type 2 diabetes. So I'm kind of gearing up for a career in diabetes or the genetics of diabetes, I think.
Yes, fantastic. And there, that's a highly that requires a high level of detail-oriented work and sort of skill in analysis. So it seems like you've got some fantastic training out of the Rich Lab, and I can't wait to see what you do in the next in your next situation. So that's going to be great. So do you want to just walk us through the paper or your work or whatever you're interested in talking about? We're interested in talking with you.
Sure, yeah. So I put together a slide presentation kind of walking through a subset of the paper because I think there's a couple of different components to it and it would have taken a bit too long to go through all of it. But so I'll go ahead and share my screen. Does that sound good?
Yeah, perfect. Go ahead. It's ready. I'm gonna.
Okay, great. So as you said, the title of the paper is Fine Mapping Ancestral, and Genomic Analyses Identify Causal Variants, Cell Types, and Drug Targets for Type 1 Diabetes. And so I guess I wanted to just first acknowledge, as you had mentioned, it's a collaborative effort. This project was a collaboration between our lab and people at UVA, as well as the John Todd lab at Oxford, University of Oxford in the UK, and was co-led by another PhD student named Jamie Inachaw, who is also a kind of a statistician, statistical geneticist, and now a statistician in industry. But and there's actually many other investigators that kind of contributed, particularly by providing samples to the study. So I'll just talk a little bit, just to give a little background about kind of what we knew about the genetic basis of T1D leading up to my thesis and kind of this work, this project in general. And then I'll talk about two components of the paper, and then kind of inspired actually a lot by some of the state of the science lectures that you have put together, talk a little bit about how this can be relevant to T1D treatment and prevention. I think because human genetics is kind of like, it's a very big problem, and sometimes it's hard to think about because it keeps demonstrating that the disease is really multifactorial and not
Yeah.
a single gene kind of disease. So I think kind of watching those conversations with some of the more experimental scientists, it's clear that it can be a little bit frustrating or confusing or maybe overwhelming to think about, like, how do we incorporate 70 genetic regions or 100 regions into a kind of unified understanding of a disease, and how could that be useful? So I'll talk briefly about that. It's not really my expertise, but it's what I'm thinking about in kind of moving forward. So
That sounds great.
yeah. So I guess the foundation of this is that your risk of developing type 1 diabetes is at least in part due to genetic factors, and that you can kind of see this in the data in a couple of different ways. One of them is, there was a recent paper, well, I guess it wasn't that recent now, but 2008, there was a New England Journal paper showing that if you follow monozygotic twins over time, so identical twins, you see that in 65% of twins with one twin having type 1 diabetes, the other one develops it by the age of 65. So there's certainly a genetic component. There's also environmental factors, which I think some of your speakers have touched on, but we focus on the genetic basis. And so I like to just briefly, because I think the audience seems is pretty broad, just kind of frame, what does it mean for a disease to be genetic, or how does genetics actually influence human traits? And just a reminder that the genome has about 3.4 billion base pairs, and it's divided across 23 chromosomes. And at almost all of those base pairs, the genetic sequence is basically identical between two people. But about 1 in every 1,000 base pairs, if you look at two people, there'll be subtle differences. So often a single nucleotide, although there's also a lot of structural variation, bigger changes. But
I love this
in general,
slide. It should be broadcast more globally, so we can all focus on what we have in common.
yeah, right. And yeah, I took this slide out, but I showed another slide. If you look at this same kind of figure, but compare across animals, like, there's a lot more differences. So if you look at within humans, we're very, very similar. But nonetheless, there's about 20 million variants that kind of commonly can differ between people in global populations. And most of these are shared across populations. So if you look at a population in one continent and then another, you're going to see both the same sets of alleles in both of those groups, but maybe at different frequencies. So kind of, like you said, we're very similar, but there are these 20 million or so places where we can differ. And so our work is to try and understand, how do these genetic variants that are common in the population, how do they shape human traits, and in particular your risk of disease? And then there also are more rare genetic variants that can have more profound effects on your risk of disease. So, Mendelian traits, for example, tend to be caused by very rare genetic variation. But the type of study that we've done in this manuscript is really aimed at trying to understand the more common, low to common frequency variants, the ones that you'll see many times in a population.
Yeah.
And so the genetic kind of basis of T1D has been of interest for, probably 50 years or so, maybe longer. And at this point, we actually do understand quite a bit about what genetic factors contribute. And the strongest genetic factors that contribute to T1D risk are HLA and insulin. And then there's these other immune regulator genes that have been identified. But it really wasn't until recently with the kind of technology developments that enabled high throughput genotyping that we were able to map really systematically the genetic variation contributing to T1D. And so in the past few decades, or 15 years or so, we've identified many, many more genetic variants that associate with T1D. When I started my PhD, it was about 60, probably a little under 60 genetic regions. And I'll show today we kind of expanded that a bit in our work. And so this concept of genome-wide association study is actually pretty straightforward. it's enabled by these microarray technologies. You basically genotype many people who have the disease and who don't, and using these high throughput genotyping tools, and then you can kind of systematically compare the frequency of genetic variants in those two groups. And using this approach, we can kind of draw these pretty systematic and robust. So if you do it again on a new set of people, it will give you a pretty similar map of the genetic basis of the disease. And so that's what we've done with type 1 diabetes. And so we get these maps, but the next step is, you hope that understanding the genetic variation that contributes to a trait will help you to understand the biology underlying the trait. And so kind of, I think in a geneticist's ideal world, or their ideal framework, is that you start with this DNA variant, and then that kind of points to genes or proteins that are important, which then helps us, leads us to cellular processes, and then kind of a way to understand how the physiology is affected by the genetic variation. Problem is, so that does work, and it has been done for both complex and rare diseases, but there are some kind of major challenges that make that more difficult than maybe kind of this idealized framework would like. So the first one is that there's something called leakage disequilibrium in human populations, or really any outbred population, where genetic variants that are close to each other travel together. So and that's just a function of meiosis and the fact that you need recombination between two variants to separate them on a chromosome. And so what that means is if you have a region, and I'll explain these plots in a second, but if you have a region of association, you have a lot of variants that all are associated with the trait, and it's very hard to distinguish which one is actually driving the association.
Right.
And so that's sort of our first major challenge in this task of taking the genetic map and inferring and understanding biology from it. And I'll explain kind of a little bit more how this works. And then the second challenge is that even if you can do a pretty good job of differentiating the variants that are likely causally related to disease, most of them don't actually overlap protein coding sequences. So it's hard to go from variant to gene. You can kind of, you can guess, or you can nominate the most likely genes mediating the effect, but to know with certainty turns out a pretty challenging task. And so just to highlight sort of in this region of association, so these plots are showing basically a stretch of chromosome, and each point is a genetic variation. So one of those, 0.1% of the genome that differs between people, and the height of the point is how strongly it is associated with T1D. And so you can see all these red ones are basically equally associated with T1D, and we don't know which one is causal. But we can take a guess that probably one of these is causal, at least one, possibly more than one. But if you look at actually how they overlap, these are the genes in this region. Many of them are in between genes, and so we call these intergenic, and then many others are intronic. So and of course they're overlapping multiple genes. So just from this map, it actually is pretty difficult to determine which of these 5 or so genes here are causally related to the disease.
Yeah, and those genes are typically found, in which cell types?
Oh yeah, so the expression of the genes across cell types is something we can look at. So I don't know about these specific genes. So I know IKZF4 is a transcription factor that's I believe important for kind of lymphocyte differentiation. I'm not sure about the other ones.
It's just curious because there's this whole interplay at the cellular level. There's the beta cell, and then there's the immune cell population, and how there's always been that whole conversation about is it murder or suicide? Is the beta cell inherently dysfunctional and the immune system just doing its job, or is the immune system going rogue? So then it's like, oh, well, which genes
Yeah.
are, that's just sort of out of curiosity, like which genes are um,
Yeah.
defective where, you know?
Right. Yeah. So people can do, so people have done analyses where you take all the all the associations and look like kind of in the region and look at all the genes and basically look for like enrichment of pathways or expression of genes in a given cell type
Yeah, like
relative
single
to
cell
other cell
seq.
types.
Mm-hmm.
Yeah, and so I think when you do that, you do tend to see that there's more enrichment in like lymphocyte genes that are expressed in lymphocytes. And we did this kind of a similar thing where we looked at chromatin accessibility, which I'll talk about in a second, but um, kind of like because you don't know which gene it is, you can kind of look at the non-coding regions and see how active they are. And there's a couple of different like epigenetic assays you can use to measure activity in a given cell type. And when we do that, we see pretty clearly that the SNPs that are associated with T1D are more often than you would expect by chance overlapping regions that are active in lymphocytes
Yeah.
and immune cell types. Although, so Kyle Galton, who you had on as a expert, so
Yeah.
his
I know.
paper,
I saw
yeah,
you guys use the T1D knowledge base too, or knowledge portal too.
yeah, we submitted our data there.
Great.
So his work kind of showed that there is some enrichment also for the islet cell, beta cell cell types,
Mm.
or the beta cells. Although I think the enrichment isn't quite as strong as if you look at the immune cell population. So, but enrichment isn't everything. So you could look at for within a given locus. I think it's harder to know. I think you can say like overall, maybe a large portion of genetic risk in T1D is driven by kind of immune factors, but within any given locus, there certainly could be effects on other cell types. So
Yeah.
um, yeah, that's one of the challenges with this kind of work is you can get kind of like global, like a sense of global trends, but then for any particular region, it's harder to nominate causal factors.
It also seems like there might be windows of accessibility or windows of activity, right? So when some genes are enriched or downregulated in certain windows in the prodrome leading up to the disease
Yeah, right.
diagnosis.
Right. So most of our like sort of maps of regulatory activity are from healthy people,
Mhm.
So it's right. So there could be, there could be features that are specific to the disease process that we just aren't seeing. So that's a good point.
You have to start, you're chipping away at a big mountain, but I mean, I think
Right.
this paper shows that you guys have made some excellent progress. So yeah, not to
Yeah.
get you off track, sorry.
Yeah, no problem. I mean, I think that's a nice feature of your series here is that it's sort of more casual and conversational. So
Yeah.
yeah, so that was all sort of background to just motivate what we did here. And so I know this is meant to be only 30 minutes, so
That's
I'll
fine. We
kind
can
of
we can
like
go a little over. It's fine. It's really interesting.
I'll try and move, yeah, relatively efficiently through what we did. So our study was kind of building on all that past work that laid the kind of foundation and also highlighted some of the challenges of understanding the genetic basis of T1D. And so what we used was there's something called the ImmunoChip, which is one of those high throughput genotyping arrays, microarray. But it specifically was designed to kind of to identify genetic variants and genetic regions associated with immune-mediated diseases. And so they basically took all the GWAS that had been done around in the like first decade of the 21st century for immune-mediated diseases and took all the top, all the genome-wide regions and kind of merged them. So they took the union of all the known immune-related traits or immune-related regions and designed a chip that really densely genotyped genetic variation in those regions.
Who did that?
And so it was a consortium. So Steve was involved, and I think Jeffrey Barrett, who is from the UK, was involved, it kind of maybe organized it, although Steve will know the history of this better than I do. But it was really a collaborative effort between a bunch of different investigators that had published these kind of first generation GWAS of immune related traits.
That's very cool.
So that's one cool thing about the genetics community is there's a lot of collaboration, I think in part because it's necessary to get the sample sizes. And so the collaborative kind of design of this chip also made it more affordable. So they were able to purchase many more of these chips than they for their studies than they would have if they had gone and just bought a commercially available array, or certainly more than if they had done whole genome sequencing. So with this sort of cost-effective targeted chip that really is enriched. So, I'll show a slide showing this, but we have this kind of idea that if the immune system is mediating effects for mediating all of these autoimmune diseases, there's probably some, we call it pleiotropy, where genetic variation that affects one trait will also affect another trait. So there's kind of this underlying hypothesis that there's be a fair amount of pleiotropy between the immune-mediated diseases, and so it would be a cost-effective way. A variant that's associated with one trait is kind of has a higher prior, if you put it in like Bayesian statistics terms, higher prior probability that would be associated with T1D. And so using this cost-effective approach, Steve and John set about, they genotyped 65,000 or about 65,000 individuals from a bunch of different studies. And we were able to use that data to kind of follow up and expand our understanding of the genetic basis of T1D. And so kind of the first thing we did was we call a discovery analysis, where you're just trying to nominate regions that, you can statistically feel pretty confident are associated with T1D. And so we analyzed all of our full data set to do this, and that included both family-based analyses. So we had trios where you had parents and affected children, and we could use certain types of statistical tools to analyze that data set. And then we also had these cases and controls that were unrelated to each other, and we could use kind of some more standard approaches to analyze those data sets. And we meta-analyzed everything together, and it basically gave us a map of the genetic regions associated with T1D. And so this is a classic human genetics plot called a Manhattan plot. And each of these alternating colors represents a human chromosome, so 1 through 22. And the height of, so each point, like I said in the previous plots, each point is a genetic variant that's in the population, and its height on the plot is how significantly it is associated with T1D. And so you can see the HLA region has the strongest associations, as we would expect, and then the insulin region, and then these other kind of known immune-mediated immune regulators, and then there's all these many other regions that are associated. So basically, any point that's above this dotted line here, we would call genome-wide significantly associated with T1D. And so that means that we feel that the statistics are sufficiently significant that we believe that is a robust association, and if you were to repeat this whole study, you would be able to replicate that. And people have shown that tends to be the case. So in fact, this study did replicate 42 out of the 43 previous regions that have been associated. So
it validated
this really
them.
is it validated them, exactly. So this kind of like gives us the skyline of T1D genetics, and it's kind of where we can start. It's sort of like the starting place for trying to understand the genetic basis of the disease, I would say. And so as you had I had just mentioned that can kind of come up is a lot of these regions, the variant that's associated with T1D is also associated with other immune-related traits. And these are that consortium that was used to design the ImmunoChip was investigators who had done studies on all of these different diseases here. And this is showing the overlap between T1D associations and these diseases. And the red indicates that the variant has the same direction of effect. So if it increases T1D risk, it also increases the risk of this other disease. and the blue actually indicates that it's the opposite. And so you can kind of see interesting trends where, like rheumatoid arthritis, all of the variants, gray means we couldn't tell based on the available data. But in the regions where we could tell, for rheumatoid arthritis, the variant that causes or increases risk of T1D also increases your risk of rheumatoid arthritis. But interestingly, for Crohn's disease, it seems like it's kind of like 50-50. Sometimes the variant has the same direction effect. Sometimes it actually has an opposite direction effect. So there could be this, there's pleiotropy, but it's not always contributing in the same way necessarily. And so we thought that was kind of interesting, and it had been previously noted, but it's always nice to kind of replicate these types of observations. And so getting into the main goal, which was to try and use this data set to do a better job of understanding what the causal variants in the region are, and thus what the causal genes and mechanisms could be, is we use this algorithm called GWAS-FM, and I won't go too detailed into this, but basically it's a method for kind of using statistics to search all the possible configurations of causal genetic variants in a region and come up with the most likely causal variants. And there's a couple of different methods to do this, but this is the one we chose to use. And one of the main kind of takeaways from this work, this part of the study, was that many of the regions have likely more than one causal variant. So you can kind of imagine there's a region with maybe one or more important immune or other genes. So genes contributing to the disease, and there are genetic variation. There's multiple genetic variants that are either both acting in different ways to regulate one of those genes, or they could be regulating actually different genes in the same region. So we call this allelic heterogeneity, where there's multiple kind of genetic variations within a region contributing to a trait. So that seems to be a major feature of T1D genetics. And this was replicated by Kyle Galton's recent paper as well. They saw the similar kind of trend of a lot of regions with multiple variants contributing. Yeah. And that's an important observation because it kind of affects how you interpret the downstream, how you follow up on things. If you wanted to dig into a region and try and understand the causal variants and their effects on, you want to take that into account. Okay, so the last part is, I guess not quite the last part, second to last part is how we try to then interpret these likely causal variants. We basically come up with sets of variants that we think are most likely causal in the T1D associated regions. And then we tried to use chromatin accessibility data. So basically an assay that helps you understand what regions of the genome are really functional and active in a cell type, how we tried to use that to understand what these variants could be doing. And so this again addresses the second challenge in kind of the genetic map to molecular mechanism challenge or project, which is that most of these variants aren't in protein coding sequences. And so the idea is if they're not in the protein coding portion of a gene, then what are they doing? And and it's kind of
$1 million question.
the $1 million question. And so people have, we have this understanding that there's these non-coding regions of the genome that maybe the formerly junk DNA that can serve these regulatory roles. And so two of these elements, I'm just kind of showing a general schematic of here's a gene and it's being transcribed into RNA. And right upstream of the gene, right upstream of the transcription start site there's something called a promoter, and that's non-coding, but it's important, it's regulatory. And then more distally, so more distant from the gene, so it can be upstream or downstream or in the introns of the genes, there's these elements called enhancers. And there's other types of regulatory elements too, but these are kind of probably the best understood ones. And so these non-coding elements are what is thought to really regulate the expression of genes across different cell types and different time points in human development. So basically we know that our variants aren't overlapping the protein coding sequence. So we want to understand, are they overlapping these otherwise functional elements that could be regulating the protein expression or the RNA expression. And so one way to do this is to look at the accessibility of the chromatin, so the DNA, at those sites for binding by things like transcription factors. And there's a couple of different assays that are used to do this. And we use something called a TAC-seq, which basically measures the accessibility of the DNA to cutting by enzymes. And that's sort of a proxy for how accessible it is in the kind of in vivo context for transcription factor binding and other regulatory functions.
Is it unspooled? Is it open for interaction with these factors, right?
Right, exactly. So the DNA kind of like, DNA is always packaged. It's not loosely floating around in the nucleus, and it's just an issue or matter of understanding to what degree is it packaged. So, really tightly packaged DNA isn't going to be, the genes from those really tightly packaged regions, the heterochromatin, isn't going to be expressed. And so if it's really tightly packaged in a given cell type that is important to the disease, or just in a given cell type is really tightly packaged, you suspect that variant is probably not having much of an effect on anything, because it's not really accessible. And so it's probably not doing things like affecting the binding of a transcription factor, because there's no transcription factors binding there anyway.
So you can rule out that region.
Yeah, exactly. You can kind of rule out that location as being relevant to at least to the function of cellular processes in that cell type. And so we did this kind of in the context of 2&D by looking at CD4 positive T cells, because we saw from other analyses that seemed like it was probably the most important cell type. And so the idea was we took variants and we showed that we wanted to see their effect on chromatin accessibility, and then their effect on gene expression, and then interpret that in the context of 2&D. So kind of briefly, our study design was we isolated T cells from patient samples. We did a TAC-seq on each of the samples, and then we systematically mapped genetic variation to chromatin accessibility. And then we could use some additional statistical tools to ask the question, is the genetic variant that's affecting chromatin accessibility likely to also be the same variant that's driving the association with 2&D? This is something called co-localization analysis. And when we did this, we basically identified 5 regions where we felt confident that, or the data was consistent with the idea that the SNP could be both affecting accessibility and affecting the trait, the 2&D risk. And so in one of these regions, I'm just showing here, this RS116 SNP, it led to less accessibility of this particular region of chromosome 14 and increased risk of 2&D. There was no EQTL, so there's no gene that seemed to be affected by this SNP. So this kind of left a gap in our hypothesis generation. But then we found 4 other regions where there were genes that we were able to kind of nominate as potentially mediating the associations. And I'm going to just end by highlighting one of these genes, or one of these regions, which encodes the gene POC2. And so in this region, we had 2 SNPs that were associated with 2&D.
So that's really interesting.
Yeah, so these 2 SNPs, we felt, based on our statistics, were the most likely to be causally related to 2&D. And so we wanted to kind of use additional data to ask whether one of these is more functional than others. And so we saw that this one on the left overlaps an enhancer, and this one overlaps a transcription start site for this SPOC2 gene, which is a transcription factor itself, actually. And we did a couple of different analyses, but in the end, the main conclusion was that this one SNP on the left, which overlaps this T cell enhancer element, appears to lead to decreased accessibility of that enhancer, and also decreased expression of the SPOC2 transcription factor. So
across all those cell types.
Yeah, so the effect appears to be relatively specific to T cells. So so that kind of, this is just showing that we did some additional analyses to kind of verify that that the effects were all kind of mediated by the same causal factor. And so, yeah, so actually the effect seems to be specific to T cells. And so that kind of led to this hypothesis that we formulated, where we felt, and this is supported by a couple of other analyses, but I didn't have time to show everything. But the idea is that the hypothesis is that the SNP leads to less binding by a transcription factor at that enhancer in T cells. And then that leads to less expression of this SPOC2 gene in T cells, and that may be the feature that mediates the association with T1D. And so this kind of hypothesis.
Oh, just to qualify, sorry, maybe I missed this. This is just in CD8s and CD4s?
Yeah, I think the effect is seen in CD8 and CD4s. It's interestingly kind of specific to unstimulated cells. So there's a lot of people have done a lot of these types of assays so if you take either CD4 or CD8 cells isolated from primary cells isolated from individuals, and then you stimulate them, for example, through the T cell receptor signaling pathway, you can see oftentimes changes in the chromatin accessibility and in the gene expression patterns.
Right. That
which
makes
is basically
sense, they have to gear up, for doing their
exactly.
business.
They have a lot to do. And so you can then actually ask, does the SNP affect the expression of the gene or the accessibility of the chromatin in the stimulated state versus the unstimulated state? And so when you do that, you see that the SNP appears to affect the expression in the unstimulated state, and also, the chromatin at this location appears to only be accessible in the unstimulated state. So there's something about stimulation that kind of shuts down this enhancer element, it seems. So that's also interesting, because it could help guide additional experiments to try and understand really more precisely how this variant functions.
Or develop more precision medications.
I think that feels further down the road, but yes, I think ultimately trying to understand all these details of like the context will be really important to understand how they could be applied to developing therapies.
Right.
So this kind of hypothesis is really the goal. Like, I think in the ideal world, the goal would be to have hypotheses like that for all regions. And so there's been, in recent years, there's been some additional, some new methods coming out using CRISPR and single cell tools that may allow us to kind of more efficiently identify these types of hypotheses for many regions. So, if we have 100 regions, let's say, that are really important for T1D risk, if we could understand and develop hypotheses and then test them systematically for many of these regions, then that may kind of move us towards a realm where all of this information is useful to understanding the disease, the basis of the disease in a specific individual. And so actually, let me I'll just quickly, we're basically at time, but I'll just quickly.
I mean, again, I'd rather, kind of see these data as part of this than just clip it. So sure.
Clip it. Okay.
We can,
so
go off if they have time, if they need to go, but I'd love to be able to share this with the wider audience.
Okay, great. Well, so then I'll finish on a brief kind of discussion of applications, because I did remember some of the speakers kind of saying, how do you think about this? Like, how do we think about a trait that is affected by, dozens or hundreds even genetic regions? How do we think about that in terms of a specific individual and treatment and therapy? So, so the first thing I think to acknowledge is that in recent years, genetic risk prediction for T1D has become useful. And this is actually not from my work. This is from Seth Sharp, who was a PhD student at Exeter, and I think will now be a postdoc with Anna Gloin at Stanford.
Very nice.
So he has done a lot of of great work from Richard Orum's group, actually, kind of showing that if you use some more sophisticated modeling approaches and these larger data sets, and I think it'll be interesting to see if he's able to use our recently published data to kind of further improve this. Same with Kyle Galton's data, that you can do a really pretty good job predicting T1D risk just based on DNA sequence alone. And that's really not the case for most other complex common diseases. And this is in large part due to the HLA factor. So the HLA risk has a really strong effect on T1D, but also could be other features of the genetic architecture. But the great thing about this is it can be useful.
Yeah, I mean, do you think these data could actually be used in the clinic? So in, some kind of like, like an infant has a heel stick, the blood is taken and it's tested for a variety of different things. Ultimately, could this be one of them? A risk prediction,
yeah.
And then, that's a patient who's followed more closely, maybe has access to,
right,
offerings of prevention bio, et cetera.
Yeah, I mean, I think I'm not a clinician, so I hesitate to comment too strongly, but I think, so one thing is that it is true that genetics isn't the only thing. So it will never be perfect. The prediction with genetics will never be diagnostic, or it will never diagnose someone with certainty. But
right.
I think it could help to stratify people, and like you said, help to identify people early that perhaps should be followed a little bit more closely. There have been research studies that have leveraged genetics, not these genetic risk scores, but leveraged HLA genetics to identify higher risk people. And actually, Steve and the group at UVA has done a pilot kind of screening program in Virginia using genetic risk prediction to identify higher risk children from a very young age, because you can do this from birth, like you said. So yeah, I mean, I don't know, we may be a ways from using it actually in the clinic, but certainly for research purposes. And I think, so what I'll just say, so the value, I think, is that you can predict someone's risk well before they have clinical symptoms. And if you use that to then kind of screen people with more precise or specific and sensitive approaches, for example, using autoantibody testing, then you can identify people in this like symptomatic window, as it has been done in some clinical trials, and that could really be useful. So I just want, so other people have way more expertise in this than I do, but just highlight that there have been these kind of prevention trials, and it seems like this teplizumab study kind of highlighted, and the history of it highlighted, that the timing of the administration does matter, because there were previous studies where they were giving this drug after diagnosis, and the effect was not as pronounced. So I think using genetics in addition to other factors. So Seth Sharp and colleagues, so their group has also published work kind of integrating the genetic risk scores with other kind of demographic features, and that further increases the predictive ability of the modeling. So I think kind of like bringing everything to bear and doing a good job of prediction may facilitate these kinds of studies. So you don't have to have quite as large of enrollment or large of enrollment for monitoring to accrue sufficient sample sizes for a well-powered clinical trial.
Definitely a great way to target, those who can benefit the most.
Exactly. And I think, and then kind of moving from how can the genetics actually not just predict, but help to inform the strategy for the preventive therapy that might be tested in a clinical trial. So this is a kind of framework that was published by Amit Kara and Sek Kitharesan. They're cardiologists, and they've kind of been leading the way in terms of using genetics, the genetics of complex traits, and in particular cardiovascular disease, in clinical settings. And they kind of highlighted this, that there's this historically this kind of idea that a person may have a disease driven by a particular pathway or process or causal factor, like a single factor. But then, and then there's this alternative kind of framework where somebody could have a disease that's driven by this sort of quantitative blend of causal factors. And they hypothesize that probably for most people, at least for a trait like cardiovascular disease, and I think T1D could be similar, you're going to have not this single pathway that's important, but actually this sort of blend. And so we may eventually be able to use some of these genetic risk prediction tools, or these genetic profiling tools, not just to predict someone's risk of disease, but also to get a sense of where they fall on the spectrum. Because I think a lot of things suggest, like, for example, the failure of the nod mouse to help us identify effective therapies, that it's probably a pretty heterogeneous disease. So there are probably people for whom the beta cell intrinsic factors are more important than for other people. And so perhaps, if we can get more efficient at mapping genetic variant or associations to causal variants in genes, we may eventually be able to use these sort of genome-wide profiles to get us a sense of the blend of pathways that are most important for disease in a given individual. And so, that's pretty futuristic, but I think
it's talking about personalized medicine, which I think is, really on everyone's mind in the larger realm, right? In the clinical space, like, okay, you cannot just treat everybody the same. So.
Yeah, exactly. And I think even before you get to the kind of clinical implementation for the general population, even in a trial, you might not expect everyone to respond to a treatment equally. So if we can do a better job at like kind of matching the people to the either immune modulating therapy or could be, a beta cell protective factor or something, then that will probably lead to more successful trials and get therapies to people faster. So
and for those that think the GWAS people and the genetics people, like I've heard on the street, oh, well, what does genetics bring to bear, and how do they really bring things to clinic? I mean, you just hit the nail on the head. This is exactly what needs to happen, is the full characterization of different subpopulations and individuals, even if we can get to there, and then addressing those with the different with the best drug for that profile. It's definitely the best way forward. I agree.
Yeah, and I think this has sort of already been done in the context of like monogenic diabetes, which is not, I would say it's not T1D, but people, I guess, my understanding is there has been some success, for example, at identifying people with MODY, and not treating them with things that won't work. And so that's like kind of, I think ideally we'll get to that point with the more complex forms of the disease. And it may take some time. And I think it will really depend on our ability to get better and more efficient at the molecular side of things, because, to map a genetic variant to kind of like a pathway or to a cell, major cell type driving the disease process, we need to be more efficient at trying to understand what the causal variants in genes are. So, but there's a lot of people working on that question, and not just people in T1D. So the whole field of human genetics, there's a lot of people across the field that are interested in this question, and major grant funding mechanisms to try and encourage that work. So I'm optimistic that there will be a lot of progress in that area. And so I guess I'll finish 5 minutes over.
No worries.
Oops. But and just thank everybody so these are all the people at UVA who mentored me and also contributed to the data generation. And then these are all the people at University of Oxford that were really involved in this project. And again, Jamie and Shaw. And then these are all the people and studies that contributed samples. So, more than 65,000 people contributed DNA to this study, which is pretty remarkable. And I'm always just grateful to those people for being willing to do that, because I'm sure it's, if nothing else, an inconvenience. So these types of large scale studies really rely on people being engaged with the research community. So
Yeah, no,
yeah.
that's fantastic. And who did T1D Exchange facilitate any of these, the access to these samples, or how did you guys access the sample data? Was it just kind of an out one by one, you put it together, the lab put it together?
Yeah, so I was not, this predated my arrival. Steve is I think, he's very, very talented at this type of work. So I don't know if he's
amazing,
listening in, but he may be able to comment on how exactly all this data came together. I think it was largely just, one at a time, collaborating with people he's worked with over the years.
The tremendous effort. He's done
yeah.
He's done so much in the field. So it's
yeah.
Thank you, Steve, if you're here.
Yeah.
On mute.
On mute, yeah.
Anyway, yeah, so this is really interesting. This is a huge amount of, samples. Thank you, Cassie. This was so interesting and fascinating work. You guys have accomplished quite a bit here, and so cool to see that this sort of, BOCT2 gene has been, identified, and interesting to see what else is found around, its functionality. So thanks again, and good luck at Michigan. You're in for, if you're heading there soon, you're in for a bit of shock from a Virginia winter to Michigan, but I'm sure you'll do great things there, and we'll keep an eye on you, and hope to see you again.